Hi, hello everyone. I think it's time. It's 4 p.m. Singapore time. So let's just start. So hello everyone and welcome to exciting webinar on cell dive, unrevealing pathways in pancreatic cancer. I'm very excited to be your moderator for today's session. I'm Isabel and I'm in charge of marketing for Leica Microsystems. Before we start, I wanted to orient you to the webinar platform we are using today. At the top right, you will see few tabs, Q&A, handouts, and polls. So please feel free to submit your questions during today's session, and we will publish them at the end of the talk. You can also download the handouts for easy reference. So today we will be exploring the possibility of spatial biology in understanding tumor-immune interactions. So first up, we have Michael Smith. So Michael completed a PhD in the genetics of DNA repair at the Columbia University, and he has a postdoctoral fellowship in genome integrity and cellular aging where he leveraged microscopy to understand the real-time dynamics of DNA homeostasis. Then we have uh, Adam Cliff, who obtained his PhD in molecular biology from Cambridge University in 2004. Before joining Leica Microsystems, Adam worked as a postdoc at the Institute of Molecular and Cells Biology of the Agency for Science, Technology and Research, ASTAR, as well as the Temasek Life Science Laboratory, and both based in Singapore. Previously, he also worked as a postdoc at the European Molecular Biology Laboratory in Germany. So this webinar will consist of a 25-minute talk by Michael. Unfortunately, he can't be there for the Q&A session, so it will be handled by Adam Cliff, right after the presentation. Okay, so let's start.
My name is Michael Smith. I'm Applications Manager for CellDive, and today I'd have the pleasure of speaking with you about the CellDive multiplex imaging solution, as well as some data that we've obtained using CellDive to look at a pancreatic ductal adenocarcinoma tissue sample. So before I get into the nitty-gritty technical details of CellDive, I'd like to take some time to talk about the types of problems that CellDive is really great for digging into, and probably the chief among those is cancer. And as you all probably know, cancer is a very common illness. 1 in 5 or 1 in 6 women will develop cancer in their lifetime. Traditional therapies such as chemotherapy and radiation can kill both cancer cells and healthy cells. Immunotherapy is a promising advance in this field, especially for treatment, but it's only about 30% effective. And so scientists still don't have a complete understanding of how cancer arises and how it progresses. And this has led to challenges in creating these new therapies and delivering them to the best possible patients. And one key aspect of that research area is the tumor microenvironment. So the tumor microenvironment is incredibly important because it takes us away of thinking of the tumor as just sort of a bag of genes or a bag of proteins, and brings the biology of the tumor into a much more full understanding, especially considering the blood vessels and other tissues that are near the tumor, which can have a tremendous impact on how that tumor progresses and what treatments will work. So by understanding the tumor microenvironment, we can more accurately predict patient outcomes, we can improve stratification of patient populations, and we can add to the understanding of patient molecular and genomic data. So how do we get to multiplex imaging? There have been methods to perform forms of multiplex imaging in the past. The most classic version of this is the traditional tissue imaging approach, which uses chromogenic staining. So in this case, FFP tissue blocks will be sectioned, and then those sections will be stained with 1 to 5 markers. And if additional sections are desired, then serial sections can be used. The problem with using serial sections is that there's a loss in resolution. In the genomic approach, however, these tissues can basically be ground up and something like RNA-seq can be performed. And that gives you a fairly complete look at all of the RNA molecules that are within that sample. But of course, the spatial information is mostly lost. So what CellDive is fundamentally is a way to kind of combine these approaches to take an FFP tissue block, section it into single tissue sections, and then perform a patented workflow, an iterative staining workflow on those tissues to look at many biomarkers, 5, 10, 15, 20, 60 plus. And CellDive is more than just an imager. It is a complete end-to-end workflow. There is, of course, the imager, but there's a software suite that helps you manage multiplex imaging studies, as well as advanced calibration techniques that make seamless stitching and high-quality images easy to obtain. And by performing this technique, you can get these beautiful layered images of multiple different biomarkers within that same tissue section without having to go back and use serial sections. And you can see here a colon cancer TMA that's been exposed to a variety of different biomarker stains. And these are all conjugated antibodies. I'll talk about that a little bit more going forward. And this is going to show you just the range and depth of information that CellDive can deliver to a researcher. So CellDive is a completely customizable, precise multiplexing tool. It can map 60 plus biomarkers from just one tissue section at a single cell level. It's customizable. You can design and configure experiments exactly how you want. It's precise. It collects the data valuable to you, and you can trust in your result. And it's proven. This is coming out of research that's over a decade old at this point. And it has 3 tested and refined technologies that have been merged into one solution. What is the CellDive multiplexing workflow? This is a very simple schematic of the overall workflow. But basically, you take these FFP tissue blocks I mentioned earlier, section them, perform a sample preparation, and that sample preparation just involves, typical IHC protocols like dewaxing. From there, these slides can be loaded into the imager. Regions of tissue that are desirable for imaging can be selected, and then off imager, a iterative staining and destaining process can begin. To perform this, basically antibodies that are tagged with conjugated dye molecules are added to the tissue 4 at a time. They're imaged, and then the dyes themselves are inactivated using a chemical dye inactivation solution, followed by a round of autofluorescence imaging to correct out the autofluorescence in the original image, and then the process will repeat 4 biomarkers at a time until the number of antibodies of interest in the study have been analyzed. Following that, the images are processed and stitched automatically, and then read out for analysis. And I just want to point out a couple of features of the overall solution that can make this work even easier for researchers. The first thing I'd like to point out is that the antibodies that are used for CellDive, you're not on your own in finding and sourcing them. We provide, along with the solution, a list of 350 commercially available validated antibodies to help you deal with the reproducibility crisis that can come from antibodies and reduce the overall time spent validating antibodies for your studies. The dye inactivation process, it comes from a patent issued in 2009. It's a chemical dye inactivation solution. It does not damage the sample. It does not remove the antibodies. There's no stripping. So it's very gentle on the tissues. They can be repeatedly stained, destained, imaged effectively indefinitely. It's been validated for use with FFP tissue and tissue microarrays, and it's compatible with cyanine-based dyes, but there are some dyes that are not classical cyanine dye chemistry that also work as well, such as Alexafluor dyes. The overall multiplexing workflow I introduced you to before, but this kind of zooms out a little bit more. What you would do from beginning to end is design your study to establish what types of tissues, what biomarkers you're going to look at, do any antibody validation, prepare your slides through that slide preparation process I mentioned earlier, design protocols within the CellDive software, add slides to those protocols and create batches in the software, and then proceed through your region selection steps. The CellDive imager has both a 2x and a 10x lens to help with this region selection process. Following that, again, the iterative staining process will be undertaken. To help you along with this process, you might imagine if you have multiple different studies or multiple different batches of slides, there's a lot of bookkeeping. The Solid Acquisition software suite includes multiplex imaging study management tools. The slides are barcoded so that the imager knows what step of the protocol they're a part of at any given time. And that's not something you have to keep track of automatically. The protocols that you write in the software, they contain all the details of the study to guide you through the workflow. And again, with that barcode reader, you're able to keep track of multiple samples and studies effortlessly. I wanted to touch on the advanced calibration image processing that occurs automatically as part of the cell dive process. There are many calibrations that we've baked into the system, over 10, such as objective centration, blank glass subtraction, flat field corrections, intensity calibrations, and more, as well as the autofluorescence I alluded to earlier, to leave you with fully corrected final stitched images of very high quality. You can select regions of any size and number that's of interest to you. The system is compatible with very large tissue areas, 20 by 45 millimeters. And from those large tissues, you can see here at the bottom, apologies, that you can subdivide that into any regions that you're interested in. The cell dive imager itself is based on a high content imaging platform. So it is a bulletproof imager for these types of automated imaging studies. It has a 5-channel capacity. So there's a channel for DAPI, which is important for a lot of the image corrections, as well as the 4 biomarker channels. It has a large imaging area. It does all of its imaging through slide, which I'll touch on in a second. As an integrated barcode reader, as I mentioned, and it's compatible with various forms of automation, such as a slide handling robot for walkaway imaging. And it contains a robust hardware autofocus module for reliable imaging. So I want to talk a little bit about ClickWell and automation. If you've done these types of multiplex studies in the past, maybe on the benchtop, you may have encountered a quite painful part of the process where the slides have to be repeatedly cover slipped prior to imaging and then decover slipped before restaining. And this can damage tissue and be quite time consuming. So as this is a major pain point for researchers, we developed a specialized slide holder for cell dive known as ClickWell. ClickWell it's a pretty simple tool. It uses 2 plastic cams to bias a slide repeatedly into place. And then as you can see in the video, an inverted plastic, something like a bathtub, is clicked into place to form a well. And into that well, you can flow your staining and dye inactivation solutions. What's cool about this is that the slide can actually be removed from the click well, and you could do staining on the benchtop or in a humidified chamber as is your preference. But because it has a reliable and repeatable slide placement, this is really amenable to automation. So with the click well, you're able to proceed through your cell dive studies in the way that works best for you. You could stain manually in the click well. You could remove the slides, like I said, and do it on the bench. You could keep slides in the ClickWell and automate with an automated pipetting system. And you could remove slides from the ClickWell and use something like an autostainer, like a Roche Ventana or a Leica Bond. And because these have a 96 well shape format, they're compatible with robotic arms for automatic loading into the cell dive imager. So if you need to increase the scale of your study, you can use multiple ClickWells to achieve that. And I just wanted to show this solution, which was created by our partners at Advanced Solutions Life Sciences in the US. This is a fully automated slide stainer and slide loading robot known as the Bioassembly Bot, the BAB. It features seamless integration with the cell dive imager. It has 3 active processing locations to reduce imager downtime, and it distributes reagents across the tissue dynamically with active rocking. It also features a cold storage for antibody preservation during the course of a staining round. So you can actually use this device to automate all of the benchtop steps that occur through the course of the cell dive process. So with that, I'd like to shift gears and talk about some scientific data that we've obtained using the cell dive solution on a pancreatic ductal adenocarcinoma. And the punchline I want you to walk away with is when you're dealing with a complex biological reality like cancer, complex questions require something like multiplex imaging and analysis because the scale of the questions that need to be answered is so high. So I'll just move on to the actual data. So like I said, I'm focusing on pancreatic cancer. Pancreatic cancer has one of the highest mortality rates of any cancer. The pancreas is surrounded by highly vascularized organs, such as the duodenum or the bile duct, and pancreatic cancer often invades and metastasizes to these organs, and also to more distant organs, such as the liver, peritoneum, lung, brain, kidney, and bone. Conventional chemotherapy plus radiation or advanced disease chemotherapy plus targeted drug therapy can lengthen survival, but even in patients that only have local disease, the 5-year survival rate can only be around 40%, demonstrating that there's a lot of need for improved therapies for all pancreatic cancer patients, including new immunotherapies. Understanding all cells in the tumor microenvironment is essential for improving cancer therapies, especially immunotherapies. Different regions of the tumor, such as the stroma, can behave differently from each other in the context of the disease, and the contributions of these regions may underlie the often quite different responses that patients display when treated with these emerging therapies. Beyond contributions from the stroma, there are subregions within the tumor that adapt to hypoxic conditions and can gain dominance by uncontrolled cell growth. Often hypoxia-adapted tumor cells are more aggressive. Here, using a small subset of biomarkers, just 5, 2 tumor, 2 vessel, 1 stroma, hypoxic regions of the tumor can be effectively classified compared to normoxic tumor. 5 biomarkers exceed the conventional methods for analysis on the same area of a slide, and it really only scratches the surface of information needed to understand aggressive tumor cells in the microenvironment. So clearly more biomarkers are needed to characterize the cell heterogeneity in the tumor microenvironment. Tumor cells in hypoxic regions have to adapt to the conditions and often activate altered metabolic pathways. So using multiplex imaging with cell dive, we can probe other biological pathways to see how these cells might be surviving in these regions and becoming more aggressive. You can see on this image here that there are marked off in blue normal pancreas, and in red there's hypoxic tumor, adjacent to unmarked, as you can see here in green, normoxic tumor. So those are the 3 tissue types we'll be focusing on today. And we have a hypothesis for our research, which is that tumor cells in hypoxic regions adapt to avoid programmed death and evade the cellular immune response. And in this hypothesis, there are some underlying questions to understand. Some of them could be, are tumor cells in hypoxic regions of the tumors adapted to the hypoxic conditions, and do they avoid cell death? Is this common in patients that don't respond to therapy? And what about the immune cell component in the tumor? To begin to address these questions, the expression of biomarkers meant to probe these questions can be analyzed using multiplex imaging and analysis. For example, to explore whether tumor cells have undergone altered metabolism to adapt in hypoxic conditions, 11 biomarkers, including stromal biomarkers for context, are used to characterize tumor cell metabolism within the tumor microenvironment. Here, individual tumor cells can be clustered based on the expression of these biomarkers. So here we start with markers that show altered metabolism, such as GLUT1. This would actually show you what regions of the tumors are undergoing hypoxia. Additionally, hypoxia normally induces apoptosis, but after continued exposure to hypoxia, selection for hypoxia-induced apoptosis-resistant cells can occur, and these cells have a more aggressive phenotype and often resist treatment. Coupling biomarkers for apoptosis in tumor cells with biomarkers for altered metabolism can help to understand the process by which cells avoid apoptosis and continue to grow, which may lead to improved treatments. So here we have classic apoptotic markers such as BAC, caspase-9, BAX. Finally, hypoxia can affect immune cell function and inflammation by modulating immune cell proliferation or through immune cell dysregulation. Understanding the immune cell contribution, hypoxic or normoxic tumor, with a possibility of restoring immune cell function in hypoxic tumors is a way to improve targeted immunotherapies. So here we're breaking down different types of immune cells. We have CD3 to look at T cells, CD20 to look at B cells, CD45 to look at a range of immune cells within this tissue. So with these biomarkers visualized on the tissue, we can use analysis software to perform region classifications to divide the tissue into normal cells, hypoxic tumor, and non-hypoxic tumor as shown here. So you can see that we took a region of the tissue. We have the important markers for determining what is hypoxic and what isn't in various colors. So we have our normoxic tumor tissue in blue. In yellow is the hypoxic tumor tissue, and in cyan is the normal tissue. And then we perform a region classification and cell segmentation using analysis software to subdivide those regions and understand more about them. So the layering of these dozens of biomarkers can help to identify differences in the tumor cells that are not obvious from just a few biomarkers. And state-of-the-art visualization of data has recently been developed for robust discovery from single-cell sequencing data. And these same algorithms can actually be used to mark biomarker expression and cell relationships using cluster analysis and UMAP. So here in this cluster diagram, each biomarker is along the x-axis, and cells are clustered based on biomarker expression similarities across the tissue. For example, based on the epithelial markers PCK26 and AE1 and the UMAP spatial relationships, we're able to identify normal pancreas up here and visualize the clusters separate from the tumor cell clusters So this would be your hypoxic tumor. Apologies, normoxic tumor. And this would be your hypoxic tumor. And the difference between them is the change in GLUT1 expression. Normoxic tumor cells will have a lower GLUT1 expression. And the number of clusters can be used to divide and further define the populations. Now, looking at apoptosis markers in these groups, we can see more breakdown. So it gives a more complete view of the tumor cells within the tumor microenvironment. So here, GLUT1 high hypoxic tumor cells have lower expression of some apoptotic markers. So here we can see the normal tissue. There are some apoptotic cells within that group, but they're clustered away from the rest of the tumor tissues. If we look at the normoxic tumor, you can see that it has a higher rate of apoptosis. And you can see in the hypoxic tumor that there is a higher rate of apoptosis. Apologies. A lower rate of apoptosis in the hypoxic cells and a higher rate of apoptosis in the normoxic tumor cells. So you can see that hypoxia does change the level of apoptosis that occurs in those tissue regions. And of course, tumor hypoxia results in a complete lack of CD3 and CD4 positive and CD3 and CD8 positive T cells in the tumor. So we can see from this clustering analysis that tumor hypoxia results in tumor cells with altered metabolism that evade apoptosis and have a general lack of immune cells present. And these would indicate the aggressiveness of the tumor. So one finding is that cancer cells in hypoxic regions of the tumor adapt to avoid apoptosis while simultaneously avoiding immune activity. But using this same data set, we can actually dig even a bit deeper to address the question of why these immune cells are inhibited from entering these hypoxic regions in the first place. First, let's just take a quick look at this table, which shows various immune cell types in the different tissue regions. You can see that in a hypoxic tumor, CD4 T cells, B cells, and CD8 T cells are sharply decreased, which recapitulates the previous analysis. And one other additional kind of interesting fact I'd like to point out that these regions are not completely devoid of immune cells. There's actually a reasonable population of what we call other immune. These are CD45 positive and then CD3, CD4, CD8, CD20 minus cells. And they don't fit into a neat category, but it was something that could be characterized in a future study. But the point remains, the majority of these classic immune cells are prevented entry into hypoxic tumor tissue. So why might that be? Using the previous data set, we can actually perform the same clustering analysis on each of the three categories of tissue. There is a lot to take in here, but this is the same kind of clustering analysis we looked at before. So like I said earlier, the individual cells are the data points and the various colors show the clusters in the same way as the previous visualization. And one thing you'll notice straight away is that in the hypoxic tissues, the number of clusters is actually much smaller because of the lack of immune cells we find in these regions. In normal tissue, there's 20 plus clusters. In hypoxic tumor, there's only about 15, 16. Now, if we perform an analysis of these clusters, we can actually establish how closely clusters associate spatially with each other. And we can extract some really useful and interesting biology. So because there's so much to kind of take apart here, I just want to focus on a single finding. So you'll see cluster 16 highlighted here at the bottom. It's actually smooth muscle actin, collagen, and CI-AIP1 positive. These are cells that are associated with a phenomenon known as desmoplasia. So desmoplasia is an increased distribution of extracellular matrix, like collagen, that can occur in PDAC tissues in response to the tumor. And these regions are associated with worse prognosis. This clustering data gives a clue as to why that might be. These desmoplastic cells in cluster 16 cluster often with clusters 5 and 8, and these represent smooth muscle actin positive stromal cells and CD45 immune cells, respectively. So what might be occurring in these tissues is that immune cells are getting bound up in collagen networks, and a hardening of the tissue might be preventing further entry. The cluster 5 cells also suggest that an EMT might be taking place, an epithelium-mesenchymal transition. So taken together with the previous data, this might be a mechanism for how apoptotic networks fail to be triggered and increased aggression of these tumor cells. So the finding here is that in hypoxic tissues, collagen deposition, desmoplasia, and EMT might underlie that lack of immune cell invasion and overall poor prognosis. Altogether, taking this data, we can see how these complex phenomena and complex questions can benefit from multiplex imaging and analysis. If multiplex imaging wasn't available, we might only be able to look at just hypoxia or just apoptotic markers or just immune cell signatures. But here, the true depth of the overall biology of the tumor becomes clear when all these areas are interrelated. And performing multiplex imaging of this kind with CellDive can reveal relationships from many different kinds of biological pathways that wouldn't be made clear using other imaging methods. So I just wanted to thank you today for your attention, and I hope to be speaking with you soon. Thank you. Bye.
Thank you. Thank you, everyone. Thank you, everyone, for your attention during the oral presentation. It was truly informative. So now it's time to address some questions from the audience. So before we start the Q&A sessions with Adam Cliff, I would like you to take 1 minute of your time to answer the poll question that will appear on your screen. So hopefully you can see the question. So do you think this multiplexing approach could help with your research? Please take 30 seconds of your time before we start the Q&A session. Great, I see a couple of people that already answered the questions. Okay, I will just wait a few seconds. And please, in the meantime, don't hesitate to key in your question in the chat, and we will select a few so that we will answer them right after. Okay, I will just wait for 10 seconds, and we can go after that to the live Q&A session. Okay, perfect. Thank you everyone for answering the questions. So now it's time for the Q&A session. So thank you, Adam, for being here. We receive a couple of questions in the chat. I will start from the first one. So how long does it take to validate an antibody and how much does it cost?
So typically you can validate an antibody in less than a week. So it would take a few hours of hands-on time, but if you have the bioassembly bot robot, that makes it a little bit quicker. So you would spend probably $1,500 to $3,000 on antibodies, so not a huge amount. We have a sequential protocol, so you first validate that it works on the FFPE tissue where we've done the antigen retrieval, so with the proprietary retrieval steps, and you can test this with primary secondary antibody first. Then we would test it again with directly labeled primary, and usually at a couple of different dye-to-protein ratios. Once you're happy that the clone you have gives a good signal and it gives a nice specific staining, you can jump straight into the process. So this is all something that's detailed in a lot of depth in the CellDive protocols as well.
Okay. Thank you. That's a very detailed answer. Second question, is it possible to image RNA with the system?
Yes, it is. So we have users that have published paper, or at least have presented posters showing that the RNA scope technique is completely compatible with CellDive. So they've done rounds of RNA in situ analysis and then gone on and done some antibody staining. So yes, it's possible to do that as well as antibody staining.
Okay, thank you very much. We have another question from Diana, sorry. So how we could design which marker first in the panel?
So it depends a lot on the kind of biological question you want to ask. We have together with the system, a lot of the documentation we give you is this validated antibody list. So that's about 350 antibodies. They're grouped based on different functions. So you could say, to begin with, we would do, for example, a couple of membrane markers just to give us outlines of the cells. So like sodium, potassium, ATPase or something. You could then look at blood vessel markers. Depending on the kind of question you wanted, you could then go and start looking at a few different immune cell markers to classify the types of immune cells you see and the types of whether the immune cells are active. You can then go into more depth and say, okay, are they activated? Are they showing exhaustion markers? You could then put on a few different markers for your particular tumor. So depending on the kind of cancer marker you're looking for, that would already give you sort of a good 15 or 20 markers. But it's quite easy to kind of mine this list and use that as a sort of source of inspiration. And we have some advice on the best ways of designing which order to do them or which colors to label the antibodies in.
Okay, perfect. Thank you. So we have another question. So do you sell the validated functional panel of antibodies?
So this is where we differ a little bit. We don't sell the antibodies. When you get the antibody list, we give you a list of the companies that sell them, the clone numbers and the catalog numbers. So you can buy them directly from the companies. You can also decide whether you want to label them yourself or whether you want to buy conjugated antibodies. We have a collaboration with Cell Signaling Technology, and we're sort of validating increasing numbers of antibodies from them, and they have a very nice conjugation service. So you can go to them, you can specify the antibodies you'd like, which colors you'd like them to be conjugated with. If you want to save a bit of money, you can do this yourself. You can also use your own antibodies. So at the moment, we don't sell any antibodies ourselves, and this is not necessarily a bad thing. If you think of the model of kind of printers and ink cartridges, having an open source system can reduce the cost of your experiments quite a lot.
Thank you. We have another question. So, hello, how does the Spatial slide holder avoid the need to open cover slip for staining?
Okay, so it's a very nice question. For CellDive, we're actually imaging with a long working distance objective through the slide. So we have a little chamber that holds the slide. There is a little lid that goes on top, and this essentially seals an area around the edge of the slide. And we can then flow liquid in and out of this. So basically there's a little hole you can pipette the liquid in and a little vent on the other side. So the slide is kept generally in media or in buffer or something just to stop it from drying out. And you can then either pipette the liquid in or out for the staining steps. And when we use the bioassembly bot, we do the staining within these little Clickwell microfluidic, or just kind of like a fluidics chamber. So the robot just comes and pipettes the liquid in. It also has a little rocker, so it will rock the whole chamber backwards and forwards a little bit to make sure we have good mixing. And this is very nice because you don't run the risk of losing sections by either decover slipping them or somehow removing these chambers between staining rounds.
Okay, I see another question in the Q&A queue. To do multiplexing at scale, do you have to automate?
Personally, I would recommend you automate it. It depends a lot on the patience of the people that are doing it. The nice thing, if we use the bioassembly bot, is that we can wait to do the staining until just before the slide goes into the imager. So while one slide is imaging, the next slide will be staining. And we can do 15 slides at once. So you would take 15 slides through a round of biomarkers in a day quite easily. And this means we can automate it both in terms of the number of probes we do, but also the number of slides you can take through at once. So this is particularly nice if you want to try and make sure that all of your slides are being treated with a particular round of antibodies at roughly the same time. So if you'd taken a very long sequential So if we took the first slide and we treated it with multiple rounds of staining, by the time you've got to the 30th or 40th slide in your study, it could be 1 or 2 months between that and the first slide. And there's always a risk that that your batch of antibody might have gone off by then. So it's not essential. The BAB robot is actually not particularly expensive, and it gives you a lot more throughput for the system. So you can leave it staining and running overnight, which is a personal preference. But if you're happy to manually load the imaging chambers into the cell dive, I know users that have done that. Or you can do the staining and we just use a plate loading robot to place the pre-stained cassettes into the imager. So we have a few different options. A lot of that would depend on your budget. You can also you can start with a completely manual system if you're working with very low throughput, and then later on add a robot or some kind of robotics to it later.
Okay, thank you very much, Adam. I can see a last question from the queue. So in the PDAC example, the final finding was still more of hypothesis. Could further interrogation of the sample be performed to continue exploring the topic?
Yes, you could do. And actually, the nice thing that we have several users that have found with the cell dive process is that you could get this far. So you could stain maybe 20 or 30 biomarkers. You can then put the slides in a glycerol buffer and just leave them in the cold room for a few months. Go and analyze the data you've got. If you realize, actually, I kind of wish we'd looked at these 2 markers as well, you can then take the same slides out and just continue the experiment after this. So you don't have to go and get fresh samples from patients. You don't have to restain all of the markers you've done. You can kind of pause the workflow, analyze it, and then come back to it again later, further down the line.
I see another questions. What type fluorescence illuminator?
So this is a wide field microscope. So we're using an LED light source for the different wavelengths. So it's nice, stable LED illumination. And we do what would be sort of Psi 2, Psi 3, Psi 5, and Psi 7. So down into the near infrared.
Okay, well, I don't see any further questions. So please, if you have any additional question, feel free to key in into the chat. We'll be happy to answer them. Okay, I don't see any further questions, so I think we can end up the session now. So thank you, everyone. Thank you to our guest speaker for sharing the expertise with us today. We really appreciate your time and insights. Also, a big thank you to our audience for attending today's webinar. We hope that you found it informative and valuable. If you have any further questions or comments, please feel free to reach out to us. Oh, I see another question. So maybe we can just answer it first before ending up the webinar. How long lifetime for LED?
This isn't something generally that light source manufacturers specify. So typically modern LED light sources for fluorescence last a very long time. So I would expect you could get a typical lifetime of a few years out of this. But this is not something that I have the numbers for. So.
Hope it answered the questions. Okay, I think we will stop now. So thank you everyone for asking questions, for your attention during today's session. Again, if you have any further questions, please feel free to just reach out to us. We will be more than happy to answer to all of your questions. You can indicate your interest in the pool that you will receive right after the webinar, or you can contact us directly by email. You can also go directly on our website if you're looking for additional information. But again, please feel free to contact us on this email address, educate@leicamicrosystem.com. Once again, thank you Thank you very much, everyone, for joining us today, and we look forward to seeing you at our next webinar session. Thank you, everyone, and have a nice day. Thank you, Adam.
Thank you, everyone. Have a good day.
Thank you, everyone. Bye.